Our final speaker of the day is Winston Timp. He is an assistant professor of biomedical engineering at Johns Hopkins University. He's a leading expert in nanopore sequencing and epigenetic analysis. Some of his recent publications have looked into direct RNA sequencing, done some Cas9 targeted nanopore sequencing, and then characterization of epigenetic modifications of RNA and DNA. So I think it's going to be a really interesting talk. Kind of as an aside, Winston was actually one of the people that got us really involved and started in sequencing, not just on Nanopore, but particularly with extraction of plant DNA. So with his work on the Save the Redwoods Foundation project with the giant sequoia and the coastal redwood, that really jump-started our move into that area. So with that, we're definitely appreciative. So I guess Winston, it's yours. Take the floor.
Thanks, Jeff, and I hope everybody can hear me. And let me say also, you got it backwards because you guys saved me. It turns out that we had a heck of a time trying to purify DNA from sequoias because of all the polyphenol compounds, and we wouldn't have been able to do it without working with circulomics. So it's been a two-way street. Thanks for the kind introduction. As said, my name is Winston Timp. I'm going to tell you today, we've heard a lot today about work on making genomes, and that is sort of the title of the symposium. But I also want to tell you about what we can do once we actually have the genome. So one of the beauties of genomics is that we don't usually want to stop there. We also want to look at like characteristics of the genome. And there has been a little bit about structural variation and changes there, but there's also a feature laying on top of the genome of the epigenome. And then there's another feature on top of that, which is what genes are expressed in the transcriptome. I'm going to try and tell you today about some of the efforts that my lab and some of our collaborators are working on towards that. So we've heard a lot today about different modes of sequencing. Today, my talk, my lab uses Nanopore, we use PacBio, we use Illumina, we use all sorts of different sequencing modalities. But today I'm going to talk to you primarily about using Oxford Nanopore sequencing. As a first conflict of interest disclosure, I do have 2 patents that have been licensed to Oxford Nanopore. Oxford Nanopore is particularly interesting to us because not only is there no theoretical limit to the the sequencing read length, but moreover, the yield is actually improving dramatically recently, and it characterizes the molecule directly. And you'll see why that's important throughout my talk, but it means that we can actually look at the DNA molecule as it's passing through the pore and characterize it, including all of its modifications and other changes to it. This is in contrast to other modes of sequencing, which primarily rely on amplification in one way or another. Or sequencing by synthesis, I guess I should say. I did also want to remark on this point. I know Calvin was talking quite a bit about sequencing and preparation, but I wanted to remark that the yield for nanopore sequencing particularly, and also from PacBio Sequel 2, but this figure is about nanopore, is improving dramatically such that the yield costs on a good run of a Prometheon. So picture on the right is a mouse genome sample that we had from tail blood from a mouse sample, actually from NHGRI. And on the left is a sample of some Chardonnay grapes. For some reason, my students wanted to go gather the DNA directly in Italy. This was before, obviously, the pandemic. I wasn't quite sure why they wanted to go and like hang out in an Italian vineyard, but they seemed excited about the prospect. But in any case, the yield and versus the costs are now commensurate with Illumina sequencing from even a NovaSeq run where it's like $25 a gigabase. So that means that long reads are now affordable. So if they're affordable, what can we do with them? Well, one of the things that we can do with long reads is we can actually look at modifications. So when I'm talking about modifications, when I'm talking about human or mammalian samples, I'm usually talking about a modification called 5-methylcytosine. So for those of you who don't remember biochemistry, your biochemistry classes, these are the structures of T, of C, and of 5-methyl-C, so pictured here in the top left of my slide. And you can see that they look sort of similar. T is different than C in that it has, T has a methyl group over here on this 5-carbon, and it has a carboxy versus a sort of an amino group on C. And 5-methyl-C sort of lives a little bit in between, right, where you have the methyl group and you have the amine instead of the carboxy. So it turns out that when a chimer is passing through a pore, if it contains a T colored here in blue, a C colored here in red, or a methyl C colored here in green on this histogram, you can actually get different currents that you're measuring as that molecule is passing through the nanopore sequencer. Because of this, we suppose that we might be able to actually call a methylation. We might be able to actually identify methylation on the molecule directly as you're sequencing. This means that if you're doing native sequencing, if you're sequencing the genomic DNA that you extract directly from the plant or animal or cell sample that you're looking at, you get the methylation information along with the other information. You get it sort of for free. And so working with our collaborator at Ontario Institute for Cancer Research, Jared Simpson, we added this module to a piece of code called nanopolish to be able to interrogate methylation. I should mention also that our friendly competitors in this regard, including Benedict Patton and Matane and Hugh, worked on a similar method, which was published back-to-back in the same issue of Nature Methods to try and characterize methylation. So many groups are working on this. Oxford Nanopore has their own tool to do it now. We are primarily continuing to work with Jared, and we use this tool, nanopolish, to try and call methylation. It works quite well, but we weren't satisfied with just looking at DNA methylation. We wanted to be able to look at methylation and chromatin accessibility at the same time. So we were inspired, so think about chromatin accessibility. What I mean by that is how open the DNA is. So, normally in your nucleus, your DNA isn't just sort of hanging out in there loose. It's wound around nucleosomes, pictured here by these green nodules. It's bound, there's certain proteins that are bound, which occlude pieces of the DNA. And Kelly et al. Back in 2012, so 8 years ago now, working with Pierre Jones, they developed a method called GNOME-seq, where they use a methyltransferase, an exogenously applied enzyme that will then decorate DNA at GPC sites. So normally mammalian methylation is at CpG sites. You can decorate DNA at GPC sites, but it'll only work at the places where it can get into the DNA. So that means at only the places where the DNA is accessible, only the regions between nucleosomes where protein is not bound to the DNA, where you get decoration of the DNA at these places. We can then read that out with the nanopore sequencer, and importantly, we can read that out at the same time as we read out CpG methylation. So a former student of mine, Isaac Lee, he's now at Pfizer, developed this method that he called NanoNoMe, where you can look at CpG methylation, as shown here on the left, and GPC methylation, as shown here on the right, to simultaneously look at CpG, to look at normal native methylation, to look at chromatin accessibility, and get the genomic information all from one single assay. And this work has recently been accepted to a journal and is now in press. A couple of sort of interesting foibles about this. If you look at the long sequencing reads we're generating here, and you look at how big the regions of accessible DNA are, you can sort of infer from these regions of accessible and inaccessible regions, you can figure out if it's a small little protein like a CTCF bound. You can figure out if it's a whole nucleosome, and the size of the inaccessible region tells you that it's sort of a mononucleosome, or if it's a dinucleosome, two nucleosomes sort of packed close together so that the enzyme can't get in between. You see sort of this bigger gap, and it bins very nicely, as you can see, to very clear and distinct different sizes. So that means that when we see using the footprint, using the area that's inaccessible to our enzyme, we can characterize if it's a protein bound, or if it's a nucleosome bound, or if it's a dinucleosome bound. We use this at known sort of CTCF, which is an important protein for DNA organization. We use this at CTCF binding motifs to interrogate and see, okay, is DNA bound or unbound? And so using our good old friend GM12878, which is a very well characterized ENCODE tier 1 cell line, we tried to look at either regions that had a CTCF peak as according to CHIP, or that did not have a CTCF peak as according to CHIP. And we were able to characterize here, as you can see, so what these different rows represent are either inaccessible in sort of orange pastel-y color, or accessible in the blue color, chromatin. And the dotted line indicates where a CTCF motif is. And this is sort of an aggregate meta plot of a bunch of reads at CTCF as a certain coordinate of chromosome 1. here on the left versus here on the right, and the dotted line shows you where the CTCF motif is, you can see that you see this little tiny inaccessible region in the middle of a larger accessible region. And we infer that this is a place where these are molecules where CTCF is bound. Then you see down below that it's a big swath of inaccessible DNA, or perhaps it has an accessible region, but the region is too large. The inaccessible region is too large, presuming that it's a nucleosome. We consider these unbound. Looking on the right, you can see we got maybe 2 reads that were that had CTCF binding, and then the 2% of the reads had CTCF binding, but then the rest of them, the vast majority, were unbound. What we think is that actually this is a quantitative measure of how many, because we're not doing any PCR, so this is actually a quantitative measure based on sampling of how many molecules in your cell sample actually had CTCF bound versus unbound. And if you instead look at CTCF ChIP-seq peaks, chromatin immunoprecipitation peaks, you see a sort of a wide variety of the fraction of bound reads. So ChIP-seq, by its nature, was not necessarily expected to be a quantitative measure. You're doing PCR. There's going to be dependent on PCR biases in the region. It's going to be dependent on lots of different peculiarities. And so we see a wide variety of fraction of bound reads with this sort of footprint measurement that coordinate with whether there's a peak there. In contrast, when we don't have a ChIP-seq peak, we generally see very, very few bound reads, suggesting that you can sort of trust if you don't see a ChIP-seq peak that probably there's nothing there. But if you do see a ChIP-seq peak, you don't necessarily know the occupancy or the strength of binding. Looking more specifically at a given region, and this is on chromosome X, the long reads give us the virtue of being able to look at the CTCF bound region. So pictured here, you can see on the left, you can see native DNA methylation. On the right, you see the GPC accessibility. On the top plot indicates sort of the bulk or aggregate data, and the bottom plots are each row indicates a different read. And we bin the reads again into either sort of active in light blue or inactive in red. And you can see that there's a CTCF ChIP-seq peak here colored in green, along with the annotation of the PIM2 gene, which is the gene that we're looking at here. I want to draw your attention to a couple of points. First of all, you can see that the promoter region in the active reads is sort of very accessible, whereas in the inactive reads, it's inaccessible. You can also see that that in the active reads, we seem to have a short inaccessible region, which we attribute again to a CTCF bound region. So on the same reads that are, 2 kb, the promoter being 2 kb away, so you wouldn't necessarily be able to see this in other ways, because there's no SNPs in this region, so you can't just phase it. We see that the promoter is accessible and CTCF is bound on those same molecules. In contrast, for the inactive reads, it's inaccessible, and we don't necessarily think that it's CTCF bound here. We just think that it's nucleosomes and heterochromatin for this other reads. I should point out again, this is chromosome X. Sort of famously, you usually have 1 active chromosome X and 1 inactive chromosome X on those of us who have 2 X chromosomes, and this is also true in GM12878. On the left, you can see that DNA methylation, largely the entire molecule seems broadly unmethylated here in the active reads, whereas in the inactive reads, the promoter is pretty methylated, but the CTCF binding region is pretty unmethylated. So just looking at methylation, we wouldn't be able to infer what's going on with CTCF here. We have to also have the accessibility information. I was just saying how there wasn't a SNP in that PIM2 region, which makes it hard to sort of phase. And so instead we're using the epigenome to phase. But using long reads, we can usually phase because we're likely to encounter a SNP when we have reads that are long enough. This means that we can phase methylation and chromatin data across pretty much the entire genome. And on GM12878, because we know the genotype there, it's a very well characterized cell line, we were able to phase the reads to either a maternal or paternal allele using WhatsApp. Over 65% of the reads were phased, which covers 86% of the genome to at least 10x coverage on each allele in our data set that we have. So we think that this represents the first methylation and chromatin phased sort of epigenome by virtue of having these long reads. But the method is straightforward to use. And I would say that there's no reason in principle why we can't do this with many other cell lines and samples going forward. Using this phased epigenome, again, we looked at chromosome X because it's so distinct and the different alleles are so strong. And we found that we can measure the methylation and chromatin accessibility on both the maternal and paternal alleles separately and look in the promoter regions. And we found that the promoter region DNA methylation for the paternal reads, generally those promoters were highly methylated. This usually corresponds to a lack of gene expression or low levels of gene expression, whereas the maternal promoter region was generally unmethylated. In contrast, accessibility wasn't so stark. We did see that the paternal GPC accessibility was maybe a little bit lower, and we saw more promoters that were accessible. But you know, we still think that it's interesting to try and dig into and interrogate this. It's also true that the accessibility might be blocked even by RNA polymerase binding there and starting to express the gene. So more study is needed to understand sort of exactly what's going on from this phased accessibility and this phased epigenome. But we think it's going to be a powerful tool going forward to try and look at allele-specific effects of the epigenome. And so I told you a lot about how, you know, it's now relatively cheap to do long read sequencing, but with the epigenome, there's a lot of noise. There's a lot of variation associated with this. And so often we want really high coverage at targeted regions that we're interested in. And so working with Oxford Nanopore, a student of mine, Tim Gilpatrick, worked to develop this sort of Cas9 enrichment method. Ida was talking about this a little bit earlier today, and I encourage you, if you didn't catch her talk, to perhaps watch it on the recording if that becomes available, and check out her paper too. Our work, we were looking primarily where she was looking at off-target effects. Our work, we were looking primarily to try and enrich for specific targeted regions. It works as follows, where we have a region of interest. We take genomic DNA that we've isolated. We dephosphorylate all the ends. We then introduce Cas9 with guide RNAs to make fresh cut sites. These fresh cut sites are phosphorylated. We then ligate on sequencing adapters, and we're able to then preferentially sequence just the regions of interest. This works pretty well. It's sort of pictured on the right, where we get relatively high coverage at, for example, GSTP1. It works with different cell lines. It works on minion. It will also even work on the flongle, which is the smaller nanopore flow cell, albeit with lower coverage. We found empirically that having multiple cut sites on either side, usually 2 or 3, dramatically improved the level of coverage and enrichment we would get and the yield. You can see here that from a panel of 10 different regions that we wanted to target, this was all done on a single flow cell. We were able to get on the order of 400 to over 1,000 in some cases coverage at our specific regions of interest. We can then use this same kind of data, because there's no PCR involved in this kind of enrichment, unlike hybridization capture methods, to interrogate specific regions deeply for DNA methylation. This is a region including the promoter region of human telomerase, which is important for different forms of cancer. Pictured at the top is what a normal thyroid sample looks like, and then the middle is an early metastasis with a homozygous ETS, which is a protein binding site, sort of up here in the promoter region. And then on the bottom is a late metastasis that has either a mutated ETS binding site on the bottom or a wild type on the top. And again, we can phase the entire read associated with that mutation site. You can see here that in the normal thyroid, we have this largely unmethylated region colored in blue for the promoter. But then as you progress through cancer between an early metastasis and a late metastasis, that methylation erodes away, and that CpG, that promoter CpG island is becoming more and more methylated as the cancer is progressing. But also interestingly, the so-called wild type allele or the normal allele of the thyroid cancer seems to have completely lost that unmethylated region. Methylation is spread throughout it almost completely, whereas the mutated allele seems to still be slightly unmethylated. So using phasing of the genomic mutation with methylation can be a powerful tool to interrogate sort of the classic Knutson's 2-hit hypothesis. You know, one hit here is occurring genomically to the ETS binding site, and then the other hit is being taken care of potentially epigenetically. It's a hypothesis about this data, but this is work that was recently published by Brittany McKelsey from Chris Umbricht's lab in collaboration with Tim and myself. We can also interrogate things like structural variation. Structural variation is hard to see, as has been discussed with Illumina sequencing. With long read sequencing, we can look at it pretty cleanly, and if we know hotspots of where we expect to see structural variants, we can target them by intelligently designing where our guide RNAs are, and then we can see both heterozygous, as pictured here in the middle with this MDA MB231 breast cancer cell line, or homozygous deletions in MCF7. We can even go out to larger sizes. So this is a picture where we tried to capture BRCA1. BRCA1 is a very long gene, actually. It's in excess of 80 kb long, and it's chock full of repetitive regions. We were inspired to look at this because Mary Claire King came to give a talk at Hopkins, and she told us how hard it was to initially look at the mapping of mutations in BRCA1 because of all the ALU elements that were all spread throughout it. And if you look at this map here of the two different alleles, with the signs and line elements, sort interspersed nucleotide repeats here, you can see that it's chock full. And so using these long reads, Tim was able to, but in combination with nice high molecular weight DNA, which we purified actually using Circulomics' method and short read eliminator, we were able to get a pretty good coverage of the entire BRCA1 region and reassemble it. We found and identified new repetitive areas that weren't actually in the canonical HT38, but that were later confirmed by deep whole genome sequencing data, both PacBio and Nanopore data on this same cell line. So we can actually interrogate and reassemble regions that might be complex or be highly repetitive using this region. It is hard, though, I'm not going to lie, to get out to sort of 80 kb with this method. And I would, the last thing I'll say on this topic is I would encourage people to look into the newer read and till methods, because I think that when you're looking at trying to capture or enrich for molecules that are, say, greater than 100 kb, you may want to instead start with nice high molecular weight DNA and use more of an adaptive sequencing method to enrich. We then tried to look at variants. Matane was speaking a little bit earlier about sort of his confidence in Nanopore variant calling. I share some of that confidence, maybe not all of it, in that I think that it is clear and easy in most cases with enough coverage, which we're getting with this targeted method, to interrogate SNVs. I will point out that different strands of Nanopore sequencing, either the positive strand or negative strand, because of how the k-mers are working, can have different sort of errors that might crop up. So one of the things that we like to do is to try and filter for reads which are supported by both strands of sequencing. When we tried to look at different variant caller tools, including just pileup with SAM tools, Claire, Medaka, and Nanopolish, we found that we were able to get actually almost all the SNPs correct with a pretty low false positive rate when we used the dual strand filter. We had a false positive rate of only one SNP with Nanopolish or Medaka, and we were able to capture 166 true positives when we used our highest coverage data of the 176 that existed in the span of our captured data. And finally, on the targeted front, we're able to actually use this method on real breast tissue, both on normal breast tissue and paired tumor tissue from the same individual. And using this and the ability that we have to phase by looking at the SNPs, we were able to identify not just mutations that were specific to cancer, as shown by these red arrows, but also mutations, but also a difference in coverage between allele 1 and allele 2 here in cancer that we attribute to a gain of copy on allele 2 for that mutated allele. So we can actually look at what copies expanding. We can even call variants de novo in cancer, in addition to other structural variant and methylation information that might be interesting. And this works on tissue. It is a bit DNA hungry. It needs about 3 micrograms to start from, but it will work on fresh frozen tissue. So that's all great, and we've been talking a lot about DNA today, but there's actually another interesting nucleic acid molecule that we all know and love, which is RNA. RNA has lots of interesting questions. RNA can fold up into lots of crazy structures that actually are able to do things. It has splicing that has different dynamics and can occur at different times as the gene's being expressed, so that just looking at the DNA sequence doesn't tell you the whole story. You have to understand how the different exons might combine to get different splicing variation. And it has not just 5-methyl-C, which I was talking about before, or other modifications which might be present in DNA. It has a cornucopia of different modifications, including modifications even to the sugar itself, to the ribose itself. And so we're really interested in trying to look at RNA. And it turns out that Oxford Nanopore has a method to directly sequence RNA, or natively sequence RNA. And we worked on this as part of a consortium, including some of our colleagues at UCSC, Nottingham, Birmingham, the Ontario Institute for Cancer Research, and UBC, to develop methods and to sort of benchmark what we can do with the direct RNA sequencing. The method basically works like this. We're usually using an adapter that goes against the poly-A strand, the poly-A homopolymer put on the end of messenger RNA by cells. We reverse transcribe. This is optional. We're not sequencing the complementary DNA sequence here. We're just reverse transcribing to remove possible secondary structure for the direct RNA and to presumably improve sort of the yield that we get. And then we ligate on the sequencing adapter. Remember, we are just going to be actually sequencing the RNA molecule, the cDNA molecule, after it's served its purpose as sort of the stake in the ground for the tomato plant to grow on, is thrown away like the trash that it is, and we just look at the RNA molecule. This is important because if you just looked at using short reads, if you just looked at parts of the RNA molecule, you might not realize exactly even what open reading frame you're looking at. Looking here at P16, looking at the ORF because of splicing changes, if you start from this exon here in blue, you'll get a very different open reading frame, and as a result, a very different peptide than if you start from this from this exon here in red, exon 1B, you get a completely different peptide depending on the splicing variation. One of the advantages, I would keep talking about phasing, but it's because I love it so much, is that you can, with long read RNA sequencing, you can also phase and even look at allele-specific isoforms. So what's pictured here is IFIH1, and what you can see here is that we have two SNPs as shown in the red boxes. This is an IGV view where each of these rows is an individual read, and the SNPs are shown in the red boxes. And if you notice over here in the green box, you can see that the paternal reads, as phased by the SNPs in the red boxes, seem to have this exon, whereas the maternal reads do not have that exon. So there's actually an allele-specific isoform present here. But we're not even limited to just looking at normal transcriptomics and isoforms. We can also look at things associated like with the poly-A tail length. You might say, well, why do I care about the poly-A tail length? Well, it's added, and the length of it might be important for different questions. So, but you may also know that homopolymers are hard to measure with Oxford Nanopore sequencing because it's hard to actually determine how long they are because you don't have nice changes in the current. It's all pretty much the same current as you're in this sort of poly-A region. So again, working with our fantastic colleague at the Ontario Institute for Cancer Research, Jared Simpson, a new module was added to nanopolish poly-A, which uses a hidden Markov model to determine the region of the poly-A tail, and then uses an estimate of how long we're spending in the poly-A tail to figure out how long it must be. When we use that, we can, on the GM12878 direct RNA data set, we can infer what the RNA poly-A tail length is. I don't know about you guys, but when I looked in textbooks learning about RNA transcription for the first time, they always show like a couple A's. I didn't realize that some of these genes have poly-A tails in excess of 200 A's long. That seemed excessive to me, but it certainly is borne out by the literature and by other blotting methods and other data. Interestingly, when we looked at specifically some of these what look like bimodal distributions here, we can see that there's isoforms which still contain introns, as sort of pictured here on the right, seem to have a longer poly-A tail than the isoforms that had that intron excised. When we looked more broadly at either protein coding transcripts or transcripts which had retained introns, we saw that this was a general trend. We think that it could be possible to use the poly-A tail length to infer sort of the RNA lifetime and how long the RNA has been hanging around, since clearly the transcripts with longer introns seem to be correlated with introns with longer poly-A tails. And we, among others, are trying to work to see if we can use it, if this is a useful measure of RNA velocity. And then finally, we can also even look at RNA modifications. This is a bit harder because there's so many different modifications that it's going to be tough to dig into. Looking at EEF2, which is a very highly expressed gene here, we plotted the electrical current at a well-known modification motif, GGACU. And what we see here is that with GGA with an in vitro transcribed direct RNA signal, we see sort of this orange distribution, which shows this sort of current that looks like this. But when we look at our native RNA GM12878 data set, we see what looks like a sort of two hump distribution here, where we see this hump, which is probably unmodified, and this larger hump over here, which we suspect is the modified current distribution. We even tested this using synthetic oligos that we generated. In the interest of time, I'll move quickly and just say that we used a carrier RNA and ligated an oligo with known modifications. And we do see that same sort of expected shift in lower current for the modification for this motif. We can even, again, look at this. This modification typically occurs in 3' UTRs of genes, and we can even use the fact that we have a long read to look at an isoform where there's exon over here and look at the distribution of the current and hence potentially the modifications shown in yellow versus the one that doesn't have that exon here shown in gray. And it looks like the one without that exon seems to be less modified, I would argue, than the one in yellow. With the exon, I would say is probably more modified, but it requires more work. So in summary, Nanopore technology is full of potential for sequencing. You can look at the genome, you can look at the epigenome, you can use targeted methods to try and get really deep sequencing at specific areas. You can use it on tissue. You can even directly look at RNA molecules, look at their poly A tails, look at their modifications. But there's still a long way to go. We still have a lot more work to really bring some of these tools and technologies to their fruition. With that, I, of course, want to acknowledge all my collaborators that have worked on this, especially shout out to Jared Simpson, Fred Sedlacek, my Nanopore Direct RNA Consortium members. The work that we've been doing with the Telomere Consortium has been thrilling and very exciting. My funding sources at NHGRI and my lab, of course, who is actually doing all the work because I just sit at my desk and let the data flow in. Thanks, and I'll try and take a couple questions.
All right, thank you very much, Winston. That was very informative. We have time for a few questions here. I've gotten several questions kind of related to the Cas9 kind of targeted enrichment. Could you comment a little bit on the benefits of doing the Cas9 targeted enrichment versus the kind of adaptive sequencing and where you might see benefits of one or the other?
Sure. So this is a great question, Jeff. I would say, if you think about, there's a couple different ways to do targeted enrichment. Even for short read or long, but really for long read, we had to develop a couple different methods. I think that hybridization-based, like solution phase hybridization capture, can work pretty well out to like 5 kb. And then beyond that, it starts to get a little trickier and trickier because you're trying to hide capture longer and longer molecules. So I think that maybe in the range up to, let's say, 10 kb, you could do hide capture. In the range from 10 kb to like 100 kb, Cas9 capture enrichment works really, really well. We get good enrichment there. One of the downsides is you have to go and design guide RNAs for each region. It's sort of like as if you were back in the days of designing primers and doing amplicons for each region you want to sequence. It's a pain. Guides work. You can use ChopChop. You can use a bunch of different tools to design guides, but you have to play with it a little bit. I think that designing the guides is a bit of a hardship. You can multiplex this pretty heavily. It can work pretty well, but once you get beyond 100 kb, we had trouble even with BRCA1. Once you get beyond 100 kb, it's a little bit harder. And that's where the read until and adaptive methods really start to shine, because if you think about, for those who aren't familiar with the adaptive sequencing methods, as you start to sequence the molecule, you base call it or otherwise align it, depending if you're using uncalled or readfish, and you say, I don't want this molecule, and you reverse the voltage on the nanopore and you kick the molecule back out. The longer the molecule is, the less time you've wasted. If you sequenced an entire like 1 megabase long molecule, that could take like an hour. So you would have wasted an hour on a molecule you didn't care about. The longer the molecule is, the more time you would have wasted had you not used adaptive sequencing. So adaptive sequencing really starts to shine as you're using longer and longer molecules. So using adaptive sequencing when paired to some of these ultra long methods that we've heard about today is really where you might start to get significant advantages, I would argue, from read until style methodologies. Does that answer your question, Jeff?
Yeah, so in that case, having, like you're saying, getting rid of really short reads or having really, really long reads is really important then. So
It is for the adaptive sequencing. I think if you're trying to do adaptive sequencing on like 5 kb long reads, you don't have enough time for the adaptive sequencing to be worth it because you still have to kick it back out, then you have to wait for another molecule to be found and start sequencing. So you don't get as much of a gain, I don't think, on small molecules as you do for long or even better, ultra long for adaptive sequencing methods. Okay. This is my intuition, and I've done some back of the envelope calculations about it, but I don't have like proof.
Okay, yeah. So we have a follow-up question, kind of related, I guess. If you're trying to kind of target a long region, say 200 kb with a Cas9 tiling approach, what size would you design your overlapping regions of interest to be with the high molecular weight DNA? Go safe with 20 kb each or try to push higher?
It's a great question, Sarah. I mean, with Cas9, you're also limited because you have to have a PAM site, right? So it would depend a lot on what the region looks like. I would try and make sure I'm going to be able to anchor so that my mappability is going to be good. Like I'm not stuck in the middle of a big repetitive area and that I have enough PAM sites to play with. But given that, I would probably go somewhere between 20 to 50. I would certainly not try and stretch to do it in 2 chunks of like 100 kilobases. I think that's just making your life more difficult than it needs to be. So I think going safe with 20 or maybe going up to the 50 is a reasonable span to target. So I would probably sit in there. That'd be my instinct.
Okay, and then we have one here, methylation. So how well are you able to distinguish, let's say, 5-methyl-C from other methylation, maybe hydroxymethyl-C? Where do you see any differences between the methylation that you detect via nanopore sequencing as opposed to bisulfite sequencing?
So these are 2 good questions. I'll sort of take them separately. First of all, methyl-C versus hydroxymethyl-C. We have had a bit of a stumbling block. My lab has had a bit of a stumbling block in really making conclusive training sets on HMC because it's hard to make all the possible k-mers with HMC and have it sit at HMC. I will say that my good friends at UCSC, their paper did look at some initial results of HMC, and I think they have a subsequent follow-up work where they've looked a little bit harder at HMC with Gaussian mixture models, and they have shown that you can, in fact, be able to distinguish methyl-C from HMC. But I don't think that there's a full-on calling method that works yet because of the lack of a systematic full k-mer training set. I think it looks like we're going to be able to tell the difference just from native signal, but it does not look like, I don't think we yet can just put it into a piece of software and get an answer.
Okay, so is that going to, so are you going to have to redo things when you go from like the R9.4 to the R10? That's
why you need a robust training set method generation, Jeff, because each pore, we're empirically determining what the current distribution looks like. And R10 is even worse because it's not a 6-mer. It's actually, I mean, it's 2 like 4-mer, 3-mers or 4-mers, but you can think about it as like a 10-mer that you have to generate. So the state space and the number of molecules that you have to generate becomes even larger. So you really need robust methods to generate these training sets and train on them. The other question about methylation from nanopore versus bisulfite sequencing is also a good one. It's something we're actually looking into right now. We've identified a couple of regions where, because of PCR bias, we think that bisulfite is getting it wrong and nanopore is getting it right. We think there are other regions where individual CGs are packed too close together and nanopore sort of smooths over them because, if you have CG, 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 and the middle one is unmethylated and the other 2 are methylated, the electrical signal, because it's k-mer based reading, might not make it so clear to get that middle CG right. And so we actually, using nanopolish, we generally call things as a cluster of CGs. And so that's a place where bisulfite might pick up something that we can miss with nanopore. I would argue that generally it's regional CG differences that we care about, but it's still a point in bisulfite's favor. And we are actually actively working on that kind of question now.
Okay, let's see here. So there was another question that I got about phasing. Can you do phasing with methylation alone?
Yes.
And if so, how kind of cell-to-cell variability affect that, as opposed to kind of using the SNPs?
Yeah. So this data here is clustered based on the epigenome alone. So it's using, chromatin and methylation. But you can pretty clearly see that if you look on these top 13 reads, they're unmethylated, and these bottom 37 reads are methylated. This region of chromosome X is sort of classic in that it's one is very heterochromatin methylated, and the other one is very accessible and unmethylated. So it's easy relatively to phase just on the epigenome there. But this agrees with our sort of existing data. There is a SNP way over here on the left side that you can't see that wasn't contained in most of the reads. And when we looked at 4 of the reads, we were able to actually phase them, and the phasing sort of agrees with the maternal-paternal agreement that we think. So short answer, yes. I would also add that some of the data Karen showed earlier today looking at DXE4 and CHM13, CHM13 being a pseudohaploid line, doesn't really have a ton of SNPs. And we were able to phase in a way that makes, I'm going to say, sense based on the data, just based on using clustering of methylation. So the short answer is, because methylation is generally correlated within itself, as you're sort of seeing here, you can phase with methylation alone in regions where you're getting sort of a systematic allele-specific effect. To be completely honest, I am showing you places where there is really strong allele-specific effects, though, on chromosome X. This isn't the case for a lot of the autosomal chromosomes. So you'd have to look for places where you had allele-specific epigenetics to be able to use that to phase.
Okay, great. Well, thank you very much. I think in order to kind of end on time, I think we will stop there. And thanks again, Winston, for a great talk.
A pleasure. Thank you.